and Bois Noir, which are...
I think it's Flavançons Doré and Bois Noir. I think it's French.
And I pronounce it
Bois Noir.
I would like to introduce
FAU-GEM.
Hello and welcome to FAU-GEM. I'm your host, I'm Laura, and today I'm here together with...
Lynn.
Hello Lynn, how are you?
I am really good. I'd like to not be here on a Saturday, but it's still fun to record the podcast with you.
Obviously. We're on episode 3 already of FAU-GEM, and I would just remind our listeners and to the new people out there who have somehow come across this podcast. We have a structure for our episodes. We will first talk a bit about the team that was behind the iGEM project that we will talk about. We will then talk about the problem they addressed, and finally we will talk about the project itself. And it was not finally, now that I think of it, because we also have a new section, which is a section in which we also talk a bit about related iGEM projects, collaborations, things like this.
Yeah.
Okay, so Lynn, do you want to present the team?
Of course I do.
Okay.
So this week's podcast is about the team from Lausanne, or from the EPFL, Ecole Polytechnique de Fédérale de Lausanne. Their project name was ViTEST, and they are the grand prize winners from 2019.
Amazing.
Yes. Their team consists of 9 members who had very different backgrounds, and actually, very surprisingly, not a lot of biologists in there.
Yeah.
It's mostly masters in chemical engineering, medical-orientated robotics, microengineering, and life sciences. But I was surprised, no biologists.
Yeah, me too. You will see later on why we were surprised that there were no biologists.
Exactly. And that's it on the team.
Yeah, perfect. Amazing. We will go over then to explain a bit the problem that they addressed. So Lynn, I will start with a question for you. Do you think you would be able to tell me how many people are living today in planet Earth?
Ooh, I think it's between 7 and 8 billion.
Very good. It's actually, it's not between, it's over 8 billion right now. 8 billion for the American numeration system. For the more European, it would be kind of like 8,000 million people in the planet. And, as you can imagine, with this high number of people in the planet, and because there, as you know, we have like some sustainable development goals, because we're a lot and we have to make everything possible to get everyone to have like a, food. Yes,
Yeah.
food. I was gonna say like a meaningful, dignified life, but also
too.
exactly
that
it was
belongs
included.
together.
Yeah, it's included. So in order to get all these people to live or to have a dignified life, you need to fulfill the sustainable, we should be able to fulfill the sustainable development goals. And one of them is
food.
Exactly, it's zero hunger. Perfect. And this is also one of the reasons why nowadays there's more and more interest in plant biotechnology research, trying to make the plants more resistant to, for example, droughts or high temperatures or even parasites.
Yeah.
Or pathogens in general. And that's what they focused, or this team from Lausanne focused on.
These plants would then be GMOs, or genetically modified organisms, because, you purposefully changed them. And I think there's some fearfulness in the general public about these GMOs, where people avoid them while grocery shopping. And I think this is a topic where science communication has kind of failed the public, because GMO is not necessarily something bad. It's where a plant has been modified to be more resistant to drought, or more resistant to a very popular pathogen, so that you have less crop loss. And more viability to feed people.
Yeah.
It doesn't mean that they're in any way bad.
Yeah.
For example, there are tomatoes that keep longer on shelf life because they have been modified this way. They taste worse. I think someone has told me that before, that they taste worse, but they just keep longer, which is great, because if we have so much food wastage already, let's not exasperate that.
Yeah.
Also, we have been genetically modifying organisms since the dawn of time, basically, since the dawn of agriculture.
Yeah,
because when we domesticate plants, we modify them through selective breeding, and that's very similar to doing it in a lab. Of course, in a lab you can do it more purposefully and not just pick the best one.
It doesn't take that much time.
Yes, exactly. But it is a very similar thing, and just because it's now a lab associated, a lot of people don't like it, which I understand, because people don't communicate correctly, or scientists especially. And I say this as a scientist, that we don't properly communicate in many cases what we're actually doing and how this changes and how this is not bad.
So going back into our topic, this team in particular, the Lausanne team, they focused on pests, in particular Phytoplasma.
Yeah.
Phytoplasma is a bacteria that is transmitted via an insect, and it's been linked with a lot of damage. I think they infect over 700 plant species worldwide.
Wow.
However, they're not really good characterized, or they're poorly characterized. I think this is something that happens to a lot of bacteria or pathogens in general that don't affect humans or animal species. People are not that interested in this. But going back to what we said earlier, it's also really important to learn about this, because maybe we can make plants be more productive, more efficient, and maybe this way we can end with world hunger.
I agree.
Yeah.
You're so right.
And also not only that, but there is not really a quick and reliable identification of this pathogen in plants. And because of this, usually farmers have to use a lot of pesticides, and as you can imagine, they're not really good.
No, for example, they can go into the groundwater, and I think you wrote in our script even that 95% of them even do not even hit the target.
Yeah.
So 95% of them don't hit the target and go somewhere else where they're not supposed to be. That's what an high off-target effect is that?
They have devastating effects also on the environment. So they focused on this Phytoplasma. And do you know one of iGEM's mottos?
Yes, it's local people solving local problems.
Exactly. And because this team was from Switzerland, they decided to focus on wine and vineyards, because they're quite important in Switzerland. They have a couple of them which are UNESCO heritage sites.
Wow.
And they also have a really popular, famous wine festival. And actually they focused more specifically on a couple of species that I think, because your French pronunciation is better than mine, maybe you can do it for me.
Okay, the first one is Flavescens doré and the second one is Bois noir.
Yeah, these are the Phytoplasma-borne diseases that some European wine growers face. And the problem with this is that they actually express the same symptoms. So the leaves kind of curled inward, they have sometimes red or yellow colored patches, and the berries of the grapes, they dry up. And it is impossible to differentiate between Flavescens doré and Bois noir.
Yeah, that's good.
Thank you. And here is where the problem actually comes from, or where the actually root of the problem is. Because the first one, Flavescens doré, it's actually highly contagious disease. There's actually not a cure for it. Whereas Bois noir is not really that contagious, and it's not a risk for a whole vineyard. Sorry. And also coming back to the pesticide problem, when you have a crop or a field that is infected, if more than 20% of the field is infected with it, then all the vines from that field, whether they are infected or not, they have to be uprooted and burned. And as you can imagine, this causes farmers and growers, cultivators, they lose a lot of money. Yeah. And this was the problem they wanted to tackle. Also, kind of related to how is it being addressed right now, before this iGEM team decided to provide a new diagnostic method. The EPO, which is the European Mediterranean Plant Organization, actually has some kind of rules that farmers have to follow, in which they say that when growers or farmers suspect that they have plants that are infected with it, they have to call the phytosanitary police and they have to send, they come and they take samples and then they send them to Agroscope, which is a center for agricultural research, and then there in this place they do the pathogen identification. And the problem with this is that between the sampling and the results, there could be up to, I don't know, I think it was like 2 weeks or something like that, which takes a lot of time. And as we said, and it's highly contagious, like maybe by the time that you get the results, you have more than 20% of your crops infected, and you lose all the crop
revenue.
Revenue. Yeah. Okay, so this was the problem they wanted to tackle or address. And now I have a question for you, Len.
Okay.
Let's imagine that in Earth 2.0, we're living in a multiverse universe now. And let's imagine that you're actually a farmer. Okay. And you actually have a vineyard. You're concerned and you would like to use a diagnostic device. What would be some characteristics that you consider are essential for a good diagnostic device?
So I would say that the results have to be pretty fast, so faster than 2 weeks.
Exactly.
That would be really nice, so you can have a fast and efficient response to save most of your crops, of course. Then what always would be nice if you don't have to give your stuff to the lab, because it's just an extra step that you have to do. It takes longer, but if I could do it at home in my nice vineyard, overlooking the mountains.
Exactly. While drinking wine.
Yes, drinking my wine.
Exactly.
If I could do it at home, that would be much easier, because I can have swift response and don't have to also contract other people for this, because it also would cost more money to send it away for a lab.
Exactly.
So that would be really awesome if I can do it myself and faster.
Exactly. And this was exactly what they wanted to do. They wanted to produce a device that allowed farmers to perform the test themselves without the need of a lab or without specialized training of any kind. And also, like as you said, time is money in this case. And if you have the response in, I don't know, like an hour or something like that, you can decide if you want to uproot and burn that plant or not. And
this also makes just removing a few plants much more viable, because you can just remove the few plants when you have detected it early. You don't have to spray the whole vineyard with pesticide.
Exactly.
So it's actually
Exactly. And so we will move now to the project, which was this diagnostic device. They called it, as we said, ViTEST. And they decided to divide the project into 5 parts. One was DNA extraction, DNA amplification, toe holes, signal generation, and one pot pure was the last module. We will explain a bit what they did on each separate part, and then we will see that combining all of these parts together creates actually like an interesting device. First of all was DNA extraction, and they wanted to see what was more efficient, either doing like the typical physical chemical DNA extraction or using a microneedle patch. Do you know what a microneedle patch is?
Yes, I do perchance.
Uh-huh.
So the difference between the normal chemical extraction is, well, if you have done that before, is where you digest the cell wall for plants and then the cell membrane to get all the DNA out of the nucleus. But for that you have to digest, it takes a while, and you have to have it at a certain temperature for the enzymes to work. Not ideal for a vineyard in the open. So they did the microneedle. It's basically, it's a very, like a surface with very small needles attached to it. And these needles penetrate through the cell wall, so you don't have to dissolve them anymore. And the DNA that's in the cell is then absorbed by these microneedles, so that stick to them, and when you take them out of the leaf again, you have the DNA already absorbed through, or absorbed just means stuck to the needles of the microneedle patch, and you don't have to digest anything anymore, and you do minimal damage to the plant. It's just a very, I would say maybe a centimeter or even less square, so it's pretty efficient.
Exactly. And they actually demonstrated this. They showed that microneedle patches are much more efficient. You can do the whole DNA extraction in less than 1 minute compared to the 1 to 2 hour physical chemical extraction, because of all of these enzymatic reactions that need some time, etc. And they also saw that the extracted DNA, they thought that if you then amplify it, it's because, maybe you don't get as much DNA as when you do like a physical chemical extraction of a whole leaf or things like this. They said that, okay, well then we just have to amplify it with PCR. And they thought about using like an amplifying method. But the problem with the PCR is you actually need a machine to do a PCR. And as you, like an owner of a vineyard, I guess that you do not own a PCR machine exactly in your home.
Also, they're expensive.
I know. That's why I guess you don't have one. I mean, not very useful, but also very expensive. So for farmers, and so they wanted to achieve 3 goals. First of all, that you don't have to use a PCR machine or some sort of heavy equipment or expensive equipment. Second, they wanted to avoid this sending to the lab step. And when they actually talked to Agroscope, and they specifically told them that when creating this device, the device has to be able to detect both flavescence dorée and bois noir at the same time.
That makes so much sense, because why do 2 tests if you can just do 1?
Exactly. And I mean, as we said, if you have like bois noir, it's not really a problem. So yeah. And last of all, it also matters that it's a fast method. And that's why they decided to do actually a variant of PCR, which is called RPA, which is recombinase polymerase amplification. So, Lynn, do you know what this RPA or this variant of PCR is? Have you heard about it?
No, I have not heard about it before, but it's actually pretty convenient, especially for this project. RPA stands for recombinase polymerase amplification, and it's basically a type of PCR, kind of similar, where they have different enzymes. So they have a recombinase and a single-stranded DNA binding protein and the strand displacing polymerase. And the advantage of the RPA is that you don't have to do the melting steps where you have to heat it up to high temperatures, because it stays in one complex all the time. And you can do it at basically 37 degrees, which is perfect if I want to do it in my vineyard.
Exactly.
Because I can do steady temperatures of 37 degrees.
In summer, you get them like...
Oh yeah, absolutely.
Definitely. So yeah, exactly. And it was also kind of important for them to recognize DNA from flavescence dorée, bois noir, and also like a third control, nothing special DNA. The third part of the project was actually the toeholds. Do you know what the toehold technology or system is?
No, I actually have not heard about it before.
Okay. Basically, a toehold is a switch. It's called like this toe hold switch.
Yeah.
And it's like a small strand of RNA that regulates the expression of a gene that is downstream.
Okay.
So basically, you just have like a strand of RNA that has like a hairpin, and it also has like there all the elements needed for the translation of RNA, like the ribosomal binding sequence, and then the start codon and everything. And the thing is, this ribosomal binding sequence and the start codon are both on the hairpin of the RNA. And you have another RNA that it's able to attach to this toe hold switch, and it's able to disintegrate, or this hairpin it becomes like a straight line. So in this case, the ribosomes can attach and they can start the translation and everything.
So it's steric hindrance, basically.
Exactly. I think it's so smart.
It is. It's amazing.
It's incredible. The fourth part of the project was the signal generation. They actually tested whether GFP or CDO, which one is better. So basically, GFP is green fluorescent protein, and it basically creates a fluorescent signal, and is usually very easily measured in a plate reader. And CDO is actually like an enzyme which is able to degrade catechol into a yellow color compound. And the advantage of this is that you can see it without the need of a plate reader or anything, and it's like so strong already.
That's amazing. And you don't need laboratory equipment and just can see it by eye.
Exactly.
That's like the gold standard of at-home tests, basically.
Exactly. And the last part of the project was the one-pot pure. This was basically what was needed is that the system, the device, should be or had to be a cell-free system, so that you do not need, for example, Ecole to produce the GFP. You do not need these genetically modified organisms because, as you know, it's not easy to get them in your house, and certainly not easy for you that you have a beautiful...
Me as a humble wine farmer.
Exactly. It's not easy for you to get them, and so it had to be a cell-free system.
That makes so much sense.
Exactly. And they decided to actually use a recombinant system. So one of the most popular systems of this kind is called PURE, which stands for Protein Synthesis Using Recombinant Elements.
Okay.
But it actually requires a solution of 36 different purified proteins, which is kind of difficult to get because when it's done, usually the PURE system, they produce each protein with strains differently, like different strains produce each protein, and this is not really handful in this situation. And so they decided to create the one pot pure, because in one pot, which means like in one flask, they will co-culture all the different strains that will produce all the different proteins for this pure or recombinant system. Mm-hmm. And so the idea is that they saw that each different step or division or module that they created, they saw that they all functioned separately, and they decided to join it. And when they joined it, they end up with this diagnostic device that was able to get the different steps. So first of all, if you have a sick plant, what's the first thing that you would do?
I stamp the leaves to get the DNA out, and then I amplify the DNA, and then I put it with the toe holds, and then I get the color, so I know if it's flavescence dorée or bois noir or nothing at all, which would be ideal, to be honest. Like
exactly,
better have nothing.
Exactly. So, Lynn, do you want to take over the last part of our episode, the brand new section?
Yes, of course. So the last section we decided to add is about iGEM connections, because we want to show how collaborative iGEM really is, how you can exchange knowledge with other teams, and maybe even get a hint, if you're interested in the topic, which teams also did a very similar thing or employed the same method. For example, this year's, or the team we presented to you today, had a collaboration with the iGEM team of Thessaly. They realized during their project that they were working with a very same or similar system, more specifically the RPA, so the
variant of PCR.
Exactly. And they did the collaboration to troubleshoot problems they were getting into or that were coming up, to discuss about the workflow and the design, which is great to have. Like, if you run into a problem and you don't know who to ask, that always sucks. But if you have another team who's doing the same competition, you can ask, hey, do you have the same problem? How can we help each other? That's honestly a great opportunity. If you are interested in the toehold system, we also found other teams that have worked on them before. For example, the iGEM team of Sastra in 2019, they employed the method, as well as the iGEM team of Aboa in 2022.
Exactly. And so, well, if you've made it this far, thank you so much for sticking around and listening. And if you have some questions, feel free to shoot them via our social medias. And
our Instagram handle is iGEM Erlangen.
And see you on the next episode.
Stay tuned
and stay curious.
Bye.
Ciao.